Hello, my name is Diana Buzas. I'm about to give a short outline of my recent publication in PNAS on the epigenetic role of the conserved FES cluster biogenesis protein DRE2 in Arabidopsis thaliana. I mentioned the word epigenetic. It is a type of heritable information stored in eukaryotic genomes which can create gene expression profiles that are robust across cell divisions. For example, a metal group covalently attached to DNA can constitute epigenetic information. Basically, it involves no change in DNA sequence. It is stably propagated via DNA replication based on daughter strand information in a semi-conservative manner, and most importantly, it can be reversible. This is either in a passive way via dilution during DNA replication in the absence of the enzymes for maintenance of the metal group, or in an active way by enzymatic removal, or, as it happens most of the time, as a combination of these two. The aim of my study was to identify new enzymes that can demethylate DNA. One such enzyme already known is a DNA glycosylase named Demeter. DNA glycosylases are a type of enzymes which normally function in DNA repair via a pathway termed base excision repair pathway. It involves the recognition of a damaged or mismatched base by a glycosylase, followed the hydrolysis of the DNA strand at the repair site, and again followed by the action of DNA polymerases and ligases to complete the repair. Demeter is part of a special glycosylase subclass. It can actually recognize and remove methylated cytosines, which then get replaced with unmethylated cytosines by the base excision repair pathway. Demeter is a particularly large enzyme, and it has a number of domains with unknown function. For example, the glycosylase domain contains binding motifs for a special type of cofactors, namely iron-sulfur cluster cofactors. These are of special importance for today's talk, and I will come back to them in a moment. Demeter demethylates DNA in a highly specific manner. First, it is limited to a single cell, a maternal gamete termed the central cell, which you see here marked in red. It appears as the red dot embedded in a massive sporphytic tissue. Second, it is limited to a very narrow temporal window, namely at the time just before fertilization takes place. And third, it has a limited number of target sites across the genome. Currently, about 50 such genes are known. The central cell divides following fertilization to generate the endosperm, a tissue appearing here as a group of red dots that will terminate while nourishing the embryo. Importantly, the embryo lineage originates from a separate fertilization event, and as a result, these two parallel lineages, the endosperm and the embryo, manifest asymmetric DNA demethylation patterns. The Demeter target genes are demethylated only in the endosperm and not in the embryo. DNA demethylation in the central cell is coupled with active gene expression, which can be monitored by a GFP reporter of a Demeter target gene, in this case one termed FWA. Let's look first at the 3 time points of the top panel. 0 days after pollination is a time when plants were emasculated to prevent fertilization and the central cell is mature. This time point monitors initial DNA demethylation in the central cell, coupled with active gene transcription and visualized by fluorescence. After pollination, at 3 days, is a stage where a lot of endosperm divisions took place, and GFP continues to monitor active gene expression from the maternal unmethylated DNA. 14 days after pollination is an endpoint for seed development, and monitors the impact of earlier gene expression. In the lower panel, we can see the phenotype of a Demeter heterozygote mutant where DNA demethylation does not take place and genes cannot be transcriptionally activated in the central cell and endosperm. Seeds that inherit Demeter mutant allele abort at the end of seed development. This phenotype demonstrates that maternal gene expression in the endosperm lineage is a biologically relevant phenomenon, and also at this stage you may be able to see which goddess gave the name of the enzyme. It was the goddess of harvest presiding over grains and fertility of the earth, Demeter. The fluorescence phenotypes inspired Tetsu Kinoshita, who thought that the lack of GFP fluorescence could be a good readout for a genetic screen. By mutagenizing an FWA-GFP line, he obtained mutants in which FWA-GFP could not be activated before or after fertilization, potentially due to a lack of activity in DNA demethylation. Two genes isolated from this screen were part of the same pathway, namely the biogenesis pathway for iron-sulfur clusters in the cytosol. DRE2 is the gene that I cloned, which encodes a protein that I'm discussing today, and it has two motifs, a generic methyltransferase motif and a motif for binding FES clusters. The other protein was NAR1, published last year in New Phytologist. Iron-sulfur clusters are ubiquitous and ancient cofactors. They are very versatile due to their redox sensing abilities. As the name suggests, their composition is very simple, iron and sulfur. However, despite being simple, when these two atoms come together, they're quite unstable, which means that dedicated machineries are needed for their assembly. Such machineries are highly conserved, and the nomenclature on this slide comes from East. Each cellular compartment has its own biogenesis pathway for these clusters, and DRE2 and NAR1 are currently placed in the cytosolic pathway, and in fact this pathway in the cytosol is known to be dependent on some components from the mitochondrial pathway. Now, to simplify these pathways, first the iron and the sulfur are immobilized from different sources, and then the sulfur needs to be reduced, in fact by the interaction between DRE2 and its partner, and then they are placed onto assembly and scaffold proteins. Finally, a multimeric scaffold complex including NAR1 transfers these clusters to recipient apoproteins. Their holoforms may act locally in the cytosol, or they may be transported in the nucleus. A number of nuclear proteins are known to be FES cluster dependent, and an emerging role for these clusters is in DNA metabolism and epigenetics. I will just mention that in Arabidopsis, mutations in all of these genes mostly result in embryo-lethal phenotypes. Now, getting back to that single cell where DNA demethylation takes place, we thought that we might have a simple hypothesis at this stage, namely that the CIA pathway may actually provide the FES clusters for Demeter to catalyze DNA demethylation in the central cell. But as it turns out, things are not as simple. Let's look at DRE2 mutant phenotype in detail. To monitor exclusively maternal specific phenotypes, we use a cross where the mutation is present only from the maternal allele and the paternal allele is wild type. As you can see, half of the ovules before fertilization and seeds after fertilization, which are actually expected to inherit the wild type allele, express FWA-GFP. The other half, expected to inherit the mutant allele, was either FWA-GFP was either inactive or seed development aborted at these early stages. Now, based on the half segregation ratio, we can conclude that DRE2 has a maternal gametophytic function. Later, the seed development was heterogeneous, with seeds aborting at different stages of development. And also, less than half of the seeds manifested a phenotype at 14 days after pollination, indicating reduced penetrance. Therefore, DRE2 mutant has hallmarks that also Demeter mutants have, including maternal gametophytic function and maternal seed phenotypes. Considering the earlier hypothesis, the next question was whether all CIA pathway mutants share Demeter hallmarks. To monitor maternal gametophytic function, we introgressed FWA-GFP reporter into all CIA mutants and examined the fluorescence at 0 days after pollination. We found that sometimes FWA-GFP appeared diminished in some of the mutants, but we could not find any ovules completely lacking GFP fluorescence in any CIA mutant except DRE2. Furthermore, when we looked at maternal seed phenotypes, we also found that the presence of a mutation from a maternal parent did not result in any significant phenotype in the seed in any of the CIA pathway mutants. We can therefore conclude that amongst CIA mutants, Demeter hallmarks are unique to DRE. Expression domain of DRE2 using GFP constructs driven by the native promoter. In the lower panel, you can see transgenics in which I also including a nuclear localization signal on these constructs. DRE2 expression was broad during reproductive phase, and it was present in the ovule and seed integuments throughout the stages. It included the central cell and the first division of the endosperm, but not later divisions, as well as the embryo. Both types of constructs were able to rescue the lethality phenotype of DRE2. DRE2 also expressed in vegetative cells, and this confocal microscopy image of root cells shows that the protein can localize, the native protein can localize to both the cytosol as well as the nucleus. The next question was, what cellular and subcellular localization of DRE2 is important for the maternal gene expression in the endosperm lineage? To answer this question, I created a set of transgenics in which ATDRE2 expression was targeted specifically to certain tissues and to the nucleus. FM1 and Diana are two promoters capable of driving specific expression to the central cell. Here I present data from 5 independent complementing transgenics and at 0 days after pollination on the left and 3 days after pollination on the right. The first 2 panels in each graph are wild type and DRE2 mutant controls. The remaining are ATDRE heterozygote plants with the indicated transgenics. Monitoring FWA-GFP at these 2 stages, you can see that both sets of constructs with and without nuclear localization signal can complement maternal gene expression in the endosperm lineage. Therefore, the answer is that ATDRE2 functions in the central cell, possibly in the nucleus, to activate maternal gene expression in the endosperm lineage. The next question was, does DRE2 regulate other maternally expressed genes in the endosperm lineage? To answer this, I used 2 approaches. In the first approach, I analyzed gene expression of 6 genes known to be Demeter targets. For this, I used 3 days after pollination seeds dissected into 2 classes, GFP plus and GFP minus, representing wild type and mutant respectively. As you can see, none of these genes can be activated in the mutant to the wild type level. This was not the case for 2 control genes that are expected not to change. Furthermore, I asked the question of whether or not DRE2 affects the methylation levels of these genes. To answer this, I optimized an assay at 3 days after pollination stage using a DNA methylation sensitive enzyme called MCRBC. In this assay, I included a pool of Demeter's mutant seeds as a control in addition to wild-type and DRE2 seeds. I designed amplicons across the region targeted for DNA demethylation by Demeter at 4 target genes. So let's follow the principle of this assay in the top panel. In the wild-type sample, DNA originating from the endosperm will be demethylated and DNA originating from the rest of the sporophytic tissue will be methylated, and this methylated DNA will be recognized by the MCRBC enzyme and digested away. On the contrary, in the DNA demethylation mutant, DNA originating from endosperm will not be unmethylated and will be digested. More of the DNA will be digested away by MCRBC, and we should be able to quantify using real-time PCR such difference. The data in the lower panel indicates exactly this situation. Namely, we recovered a lot less DNA from both Demeter and DRE2 comparing to wild-type at the regions which are methylated, but not at the one FWA region which is not methylated. In addition to this molecular approach, I next used a genetic approach to check the interaction between DRE2 and a protein expected to have the antagonistic activity, namely maintenance DNA methylation MET1. For this, I constructed double heterozygote mutants. Looking again at FWA-GFP at the 2 stages which you are familiar with by now, 0 days after pollination and 3 days after pollination, please notice that in the proportion of ovules and seeds expected to inherit both alleles, the fluorescence is restored to wild type. This result indicates genetic suppression. Therefore, we can conclude that ATDRE2 and MET1 have antagonistic effects on DNA methylation. This brings us to 2 final conclusions. First, there is no genetic evidence to support a CIA function upstream of Demeter in the central cell. And second, ATDRE2 acts in the same spatial temporal window with Demeter at Demeter target genes and has activity towards DNA demethylation. This is the first report of an epigenetic role for DRE2 in any organism. There are currently numerous possible mechanisms for how DRE may perform its epigenetic role. To give only 2 elements from my current hypothesis towards elucidating such mechanism, first, it is possible that ATDRE2 may inhibit DNA maintenance methyltransferase activity via its SAM domain, or it may also be possible that DRE2 may provide the FES cluster for Demeter, but in a manner that is CIA pathway independent. Finally, the acknowledgements. This project was hosted at 3 institutions in Japan. These were Nara Institute of Science and Technology, Nagahama Bio, and Yokohama City University. I am grateful to the Japanese Society for Promotion of Science for offering me a fellowship, and to Tetsuo Kinoshita and his lab members Miyuki Nakamura, Yukiko Sujimoto, Yuki Kinoshita, Yoko Ikeda, Akemi Ono, and Takayuki Onishi. Thank you for your attention.